Hello, my dear students. Today we are going to discuss some top 10 concepts of the chapter Biomolecules. So, I have already made a format of these concepts. So, these are more of facts than concepts, I may say, but we're going to call them concepts for now because we need to learn them in a systematic way. We need to learn them in a proper way so that we'll remember them forever. So let's start from our first concept. The first concept I have kept here, that is the important polysaccharides. Okay, for important polysaccharides. So if we talk about your book, some important polysaccharides include starch, glycogen, cellulose, and chitin. There's also another one, inulin. But we need to discuss these four importantly. So first of all, you may be asked, what is this polysaccharide made up of? So talking about starch, we all know that starch is made up of alpha glucose. Talking about glycogen, the answer again will be alpha glucose. But talking about cellulose, the answer will be beta glucose. So cellulose is made up of beta glucose, and chitin beta and acetyl glucosamine. All of these are homopolysaccharides, one should note. Then, next thing that may be asked, both starch and glucose are storage polysaccharides. So, starch is stored in plants, glycogen is stored by these animals, cellulose is a structural polysaccharide, is found in cell wall, and chitin is also a structural polysaccharide, it's found in cell wall of the fungi and exoskeleton of the arthropods. Then, important thing that is usually asked is about the helix formation. So helix formation is present. Helix formation by the glucose chains is present in glycogen as well, but helices or helix formation is absent in case of cellulose, and it is also absent in case of the chitin. So no helix in cellulose and chitin. The next thing that we may be asked is about the color with iodine. So we must all remember that this starch gives blue color with iodine, which is often asked. Blue color with iodine. This glycogen gives reddish brown color with iodine. Reddish brown color with iodine. And if we talk about cellulose, there will be no color. And if we talk about chitin, there will be no color. So this is the first key concept about the polysaccharides. One should always remember this thing. In 2023 NEET, it was asked about cellulose, that does it form helix? No. Does it give any color? No. So now we have to discuss the second concept, which is about the structure of proteins. So we must do it in a precise way. There are four structures of proteins. There are four levels of structure of protein. One is so-called primary structure. Second is so-called secondary structure. Third is so-called tertiary structure. And fourth is so-called quaternary structure. We are going to discuss them in a very precise way so that our questions are answered. So talking about primary structure. So a lot of times it is asked, what is primary structure? So primary structure is nothing but sequence of amino acids in a polypeptide chain. Sequence of amino acids in a polypeptide chain. In a polypeptide chain. So sequence of amino acids in a polypeptide chain is what we call as primary structure. And the bonds involved in it between the amino acids, they are peptide bonds. One should always remember. If we talk about secondary structure, it is folding of primary structure. It is folding of primary structure due to which bonds? Due to hydrogen bonds. One must always remember. And we have two types of primary secondary structure. One is alpha helix, which is more common, and second is beta pleated sheet, which is relatively less common. Then we may be asked about tertiary structure, which is always we should always remember it is folding of secondary structure. secondary structures. Folding of secondary structure due to a variety of bonds, which may include disulfide bonds, van der Waals bonds, hydrophobic bonds, electrostatic bonds, etc. And one must always remember it makes the protein functional. This structure makes protein functional. One must always remember. And quaternary structure, it is only present in multimeric proteins. It is the arrangement of the subunits with respect to each other. And one example of this multimeric protein is hemoglobin, which is having 4 subunits. And arrangement of those subunits with respect to each other is what we call as quaternary structure. So we are done with the concept number 2. Let's go into concept number 3. Concept number 3 is about the phospholipids. Phospholipids are special type of lipids which are found in our cell membranes and a lot of other tissues, other sites. So the most common phospholipids are glycerophospholipids. Most important ones are glycerophospholipids. And in these glycerophospholipids, we have a triglyceride whose third fatty acid is replaced by phosphoric acid. So this one is a diglyceride, and if we had a third fatty acid, we get a triglyceride, but we want to replace that third fatty acid with phosphoric acid like this, and this species is our phospholipid. One must always remember this R is hydrophobic, this R2 is hydrophobic, but this phosphorus is hydrophilic. So these are hydrophobic tails. These are hydrophobic tails. These are hydrophobic tails of this phospholipid, and this is hydrophilic head of this phospholipid molecule. This is hydrophilic head of this phospholipid molecule, and we represent it like this. The upper one is head and lower two are tails. And such a molecule which is having hydrophilic as well as hydrophobic part is called as amphipathic molecule. So our phospholipid is an amphipathic molecule. We must always remember this. Now, going to our concept number 4, which is very, very important about the formation of nucleotides. One must always remember that in a nucleotide, the central molecule is a sugar. So we have this sugar is a central molecule. This is 1 dash carbon of the sugar, 2 dash carbon, 3 dash carbon, and 4 dash carbon. 1 dash carbon of the sugar forms the bond with nitrogenous base, and that bond is called as NC glycosidic bond. That is called as NC glycosidic bond. And we have the 5 dash carbon of the sugar. That 5 dash carbon forms a phosphoester bond with phosphoric acid. A phosphoester bond with phosphoric acid. So this thing is phosphoester bond. This thing is phosphoester bond. So a nitrogenous base attached to 1 dash carbon of sugar and a phosphoric acid attached to 5 dash carbon of sugar makes our nucleotide. Now if it is ribose sugar, it becomes a ribonucleotide, and if we have a hydrogen at carbon number 2, then it becomes deoxyribose sugar. One must always look at the components. 3 components. This is phosphate group, this is pentose sugar, and this is nitrogenous base. And combination of sugar and nitrogenous base is also called as nucleoside. That's also called as nucleoside. One must always remember this thing. Okay. The next concept is related to the enzymes. So enzyme types. So we have 2 types of enzymes. One is called as simple enzymes, and the others are called as conjugated enzymes. One must always remember this thing. Simple enzymes are just made up of protein. They are only made up of protein. So they are made up of protein only. And if we talk about conjugated enzymes have two parts. Protein part, which is called as apoenzyme, which is called as apoenzyme, and non-protein part, which is called as cofactor, which is called as cofactor. And always remember, we have three types of cofactors. We have coenzymes, we have prosthetic groups, and we have metalines. And remember, coenzymes are organic cofactors, but they are loosely bound with the enzyme. Prosthetic groups are organic cofactors, but they are tightly bound with the enzyme. And metalines, they are always inorganic in nature. Okay, now remember, example of coenzyme is NAD positive, nicotinamide adenine dinucleotide. It is a coenzyme of dehydrogenases. Example of prosthetic group is heme, which is a prosthetic group of catalase. An example of metalines is zinc 2 positive, which is a metaline cofactor of carboxypeptidase, carbonic anhydrase, and many other enzymes. So remember this thing, two types of enzymes, simple and conjugated. And simple enzymes are just made up of protein. Conjugated enzymes are just made up of protein part and non-protein part. Protein part is called apoenzyme, and non-protein part is called cofactor. We have three types of cofactors: coenzymes, prosthetic groups, and metalines. Coenzymes are organic, loosely bound cofactors, for example, NAD positive, and prosthetic groups are organic, tightly bound cofactors, for example, heme. And metalines are inorganic cofactors, for example, zinc for carboxypeptidase and carbonic anhydrase. Now, the factors affecting enzyme action. There are three main factors. One is pH, second is temperature, and third is substrate concentration. And we must know the graphs. For pH, the graph of velocity versus pH will be like this, where you will see this pH gives maximum activity. This is called as optimum pH. This is called as optimum pH. Same type of graph is obtained when you plot against the temperature. And here you will be getting the optimum temperature for the enzyme where the activity is maximum. But in case of substrate concentration versus enzyme, you will get a graph like this, where enzyme is increasing initially in linear. With increasing substrate concentration, the enzyme activity is increasing in linear fashion, but ultimately it is reaching a maximum, where the graph becomes plateau. This maximum velocity achieved is called as Vmax. Actually, at Vmax, all the enzymes have been occupied, all the active sites of the enzymes have been occupied. Now, one thing you must note, if we take Vmax by 2 and plot the substrate concentration with respect to that half maximum velocity, that's known as Km, or Michael-Menten constant. Michael-Menten constant. And we must always remember the affinity of enzyme for the substrate is inversely proportional to Km. That means an enzyme which is having more Km has less affinity for the substrate, and an enzyme which is having less Km has more affinity for the substrate. Then another topic is about the enzyme inhibition. Enzyme inhibition is decrease in activity of enzyme due to presence of substance called inhibitor. Now we have two types of enzyme inhibition. We have a type called competitive inhibition. We have another type called non-competitive inhibition, which is usually irreversible. In case of competitive inhibition, the inhibitor just looks like the substrate. So they both compete for the active site. But in case of non-competitive inhibition, substrate and inhibitor have no resemblance. This is the substrate and this is the inhibitor. So inhibitor binds to a different site which is called as allosteric site. Allosteric site. Okay. Example of this competitive inhibition is inhibition of succinate dehydrogenase. Succinate dehydrogenase by an by a substance called malonate. By a substance called malonate. By a substance called malonate, which looks just like succinate. An enzyme of example of non-competitive inhibition is inhibition of cytochrome oxidase by an ion called cyanide, Cn negative, by cyanide ion. Okay, then let's go to concept number 7, which is talking about essential amino acids. So you must know there are 10 essential amino acids, and we remember them by the word, by the mnemonic AV Hill MPTT, where A stands for arginine, V stands for valine, H stands for histidine, I stands for isoleucine, L stands for lysine, L stands for leucine, M stands for methionine, P stands for phenylalanine, T stands for threonine, and T stands for tryptophan. These are the 10 essential amino acids you must never forget. Then we have the percentage composition of cell, where you should note that cell has maximum amount of the composition of the cell, about 60%, more than 60% is water. Water is the highest, about 60%. Then we have proteins, then we have nucleic acids, then we have carbohydrates, then we have lipids, and then we have ions. So we just remember the middle four by mnemonic Papanai Carla. So proteins greater than nucleic acids, greater than carbs, greater than lipids. And before proteins, we have the water, and after lipids, we have the ions. Okay. Then there's another molecule called cholesterol, which we should know. The most important thing about cholesterol is its formula, which is it's a type of steroid found in animals. It is basically a sterol with an alcohol group, and its formula is C27H46O. The structure looks just like this. We have three hexagonal rings fused to a pentagonal ring, and there are two carbons outside the ring. And we must also remember at carbon number 17 we have a hydrocarbon chain, R chain, and we must also note this R chain has 8 carbons, so it may be drawn with 8 carbons. Now important, at carbon number 3 we have an OH group, and carbon number 4 and 5 and 6, sorry, we have a double bond. This is how you identify the cholesterol molecule. A hydroxy group at carbon number 3, a double bond at carbon number 5 and 6, and a 8 carbon hydrocarbon chain at carbon number 17. You can just look and remember OH, double bond, and R. This is what will help you to identify the cholesterol molecule. And last but not the least, DNA double helix model, which was given by Watson and Crick. So I can say Watson and Crick's DNA double helix model. And I can say this model looks somewhat like this, and you must note some important points from this model. Some important points include the first point being DNA is a duplex molecule. That means it has two strands. This is 1, this is 2. And second, the strands are anti-parallel. The strands are anti-parallel, which means if this is 5 dash, this will be 3 dash. So one strand runs in 5 dash 3 dash, other runs in 3 dash to 5 dash. So anti-parallel. 3 dash 5 dash is one strand, so there will be 5 dash to 3 dash. Okay. Now third point, the DNA forms helix, which is right-handed helix. The two chains form right-handed helix. The fourth point being the backbone of each strand. Backbone of each strand is formed by sugar and phosphate, and nitrogenous bases project inside. And number 4, the helix is stabilized by hydrogen bonds. So adenine and thymine form 2, while guanine and cytosine form 3 hydrogen bonds. The sixth point is there are 10 nucleotides or base pairs per turn of the helix, and pitch of the helix is 34 angstrom nits, which is calculated by distance between the two base pairs, which is 3.4 angstroms into 10. And each base pair turns by an angle of 36 degree, which can be easily calculated by 360 divided by 10. 360 is the total angle and 10 are the nucleotides per turn. There are many more points or postulates of this double helix model, but I shall not discuss them because I want this video to be precise. So thank you. I hope you learned these 10 concepts precisely and accurately. Thank you very much.